Pleasure. Okay, we have started the recording, of course. Okay, just telling everyone that we have started the recording. I see that Professor Dirks has entered the meeting, I believe.
Yes.
Okay.
Good morning or afternoon. Hi, everyone. Good evening.
How are you? Nice to see you.
How are you? Nice
to see all of you. How are you doing?
That's good.
Thank you. Thank
How
you.
are you?
Thank you. Great.
I'm very well. Yeah, a few familiar faces and names here, which is great.
Yes, I think last
How's
time
everyone doing?
it's back to 2019, right?
Yeah, I was just thinking about it. It's like it's been like 5 years almost now. It's incredible. Yeah, few things have happened in between. But yeah, it was 2019 here. Yeah, you were just visiting us. It was what, June 2019, I think.
Yeah, it's June. It's in June.
Yeah, so it's a long time. Now it's good to see everyone here.
Yeah.
And Hamed, so just around the corner, pretty much, just London. I could fly over and be there in about an hour.
Yeah, and especially today is a really good weather if you want to come, because we have seen sun after some long time.
Yeah, finally. Yeah. Hamed, when's the last time we met? I think it was a PhD defense 20 years ago.
Yes, it was my PhD defense. Exactly. It was 2020. When was that? 2019, I believe. Right.
Exciting year.
Oh,
yeah, very long
also
ago.
5
Yeah.
You
years
were
ago.
also at the conference, Hamed, right? You went to England.
Yes.
Yeah,
I was in the conference too. So we met, all of us. We met in was it in Bremen?
Yeah, Idelsen, close to Bremen. So this small like castle, put it that way.
Yeah, very nice place indeed. Yeah.
Yeah, absolutely.
So how you been doing? Everything good there?
Oh yeah, that's fantastic. Yeah, I'm doing very well. Our son is now 2.5 years, almost 3. So keeps us busy, picking up and returning from Kita and all these things. Dorothea is now professor here at Hochschule as well.
Yeah, I heard that. I noticed that. Okay, that's good. So both of you are very close now.
We're safe. I can see her office from like Stuttgart times, like where her office was just.
Yes, I remember that. Yeah.
So we're very lucky that worked out. So we're quite safe here in terms of job perspective. Yeah, no, that's I'm very happy. My group's doing well, and you settled in well in London. I've seen so.
Yes, quite well indeed. Yes, in the last
output.
1.5 years.
Every Nature paper seems to have Hamed Rajabi on it right now. So.
I won't say that, but thank you.
No, congratulations. That's fantastic.
Thank you so much. Jan-Henning, would you like to share your presentation? Just quickly check whether everything is working properly.
So I think we have to. Yeah, thank you very much. Let me quickly share this. Let me see what's the best way. I'm going to try this way of sharing it. So I can see you and you can see my screen.
Amazing. Yeah, perfect. Wow, very beautiful.
Check, done. Yeah, I was very lucky. I had a very talented student, Vera, who was excellent in drawing insects, and she combined scientific knowledge, just and expertise with a very good artistic talent.
I can see that.
So this is just made up things, but she also illustrated some textbooks where she went to the museum and did some proper stuff. And this was just going crazy for the textbook, like for the title.
I see. Yeah, but I see in the middle a stag beetle. So okay, but combination of many things. But by the way, what Karen is doing? Karen
she's got a proper job now.
Okay.
She decided to leave. She finished her PhD, which is all fantastic. She did a great job, and she's a fantastic PhD. And she started her job 1.5 years ago in Bremerhaven. She's basically project leader in a scientific lab, which is like an institute which is associated to the University of Applied Sciences, but she's not pursuing the academic career.
Okay, yeah. I guess that's better.
I think she was feeling. I fully support her decision. It was better for her.
Staying in academia is not, it's. I don't know. I have also sometimes conflicting feelings, but
yeah, we've been there.
My dream, but yes.
We've all been there now, so I'm very happy I think she found a good job. Last thing I heard, she was very happy. We got the most important parts of her PhD published, so we're going to talk about this a bit today. So she did a very great job, and no, she was she's now a free person.
Yeah, exactly. I have seen some of the papers, really good ones indeed.
Thank you.
especially the last one. Was it in interface, if I'm right?
The
Or
proceedings one on hypergravity.
yeah. Anyway, very and indeed very fascinating job. The one that you published on the centrifuge, indeed.
Yes, the James Bond centrifuge paper.
Yes.
Yeah, it's proceedings Royal Society B.
Yes.
So I'm going to show this.
Yes.
Yeah, so there was no, that it took us some time. Just we had like Karen, just she already basically left it, so I had to write it up to some part myself, which is always a bit time consuming. But no, I'm very happy that we got this paper out. It's, you remember I started that when you were visiting us in Stuttgart when it was like
Yeah. Exactly.
2012, 13?
It was 2014.
14. Yeah, wow.
Yes.
That's 10
Yes.
years ago. That's 10 years.
Yes. So we were working together before I come to Germany. So I
Yeah.
guess 2 years before that. So about 12 years ago, we started working on this fracture paper, if you remember.
Yeah.
I guess it was 2014 that the paper was published in Journal of Biomechanics.
Yeah.
Yeah.
Long time.
Long ago. So yeah.
We were both young at that time, young and full of hope. Fantastic. No,
Yeah.
that's really good. No, so that, yeah, so yeah, I just remember the time we built up a centrifuge in this basement of the Max Planck Institute.
Yes.
And it looked like a zombie movie somehow. And now it's all finished, and it is the report. We have plenty of more ideas what to do with it, so we're currently applying for funding to convince the DFG to put more things into the centrifuge.
I see. Perfect. That's really good.
Yeah.
I guess that the main research body there in Germany is DFG, right? So as far as I remember, it was.
Yeah, it's for fundamental science. That's like most of my money comes from the DFG. The BMBF is also some part of the more applied research that I do. The biomimetic stuff actually is paid by the BMBF, the German Ministry of Education and Research. So that's like more they have more like some calls for funding for a specific subject. So maybe they call for biomimetic funding, or they have something, whatever, like battery research. So depending on the current interest of the government, they set aside some money, and that's managed by the BMBF. But usually you have companies aboard. So we have, for example, we're working together with Airbus on this project. But you're right, the DFG is the main research. It's like the EPSRC or BBSRC, but it's more open to fundamental stuff. So I'm a big, I like it a lot, because you can do crazy fundamental stuff. This has to be fundamentally sound, no application, no benefit for whatever society or engineering. Just if it's a great project, and you can convince the reviewers, then they fund you.
Yes. Have you been applying for European fundings too?
Actually, not yet. No. So
because from this year on, again, we can basically UK researchers can apply again.
Yeah, I heard that now you're out of the hall of shame. You can join the club again. It's so ridiculous. It's weird. Yeah, but so if you have anything, I'm more than happy. Like this. So European funding is something I don't have much experience with it, but usually it's good to have collaboration partners, many countries. So
absolutely, yes. There are many things indeed. So maybe we can discuss after.
Anytime, yeah.
Anytime.
No, for first, it's like I'm quite happy with the DFG funding. The European funding usually has some specific cause, or from my experience. So I didn't find any matches so far, and sometimes they have like very small countries, like where it's hard to find someone in this specific small country who does something similar to insect biomechanics. So but it's good that the UK joins again, so we can
exactly.
Apply again, David and you and then Walter Federle. Have you been to Cambridge, and have you met Walter Federle actually?
Not really. I haven't even met. I'm writing emails time to time to David Labonte.
Yeah, I heard.
We had a few plans to meet each other. He coming. Indeed, I got a very small grant collaborating with him, and we were planning since I don't know the time that I came to the UK to meet, but we never found a chance to meet. You know, I'm also a little bit having
I
huge
know you.
yeah.
dragging you out of the lab.
Yeah, it's not.
And you're the only person like I know who responds so fast to emails. Like in the middle of the night, I was working with Dorothea. Usually we work quite late in the evenings when Valentine is in bed, and so I was like saying, "Oh, I'm sending Hamed an email." Like, bam! Oh yeah, he's the only one. He's the only one replying at 3 o'clock in the night. Like,
Yeah, that's very right. Especially David is also the same. So whenever that I write him, he comes back to me very quickly indeed.
Yeah, I usually WhatsApp him, so I know.
Really?
But he's super fast. That's true.
Yes, and quite successful. He has a very nice team. He has
Oh
got
yeah.
interesting grants.
I closely follow his, besides my personal interest, but many of his students are former students of Bremen.
Yes.
So Fabian,
Yes.
Hendrik, you name it, they all have all been here before.
Yes.
So like I'm always very happy when I see that, wow, they grow up so fast. I just feel like just supervised their bachelor's and now they have finished their PhD.
Yeah.
So that's really it's really amazing to see that happening. So yeah. So we also work together on a few bachelor's and master, sometimes master, but I think mostly bachelor's projects.
I see. Yeah. That's really good.
I dare to ask, are you coming to SEB this year, or do we have, or are you sending someone at least?
Not really. No, that's a very busy time for us, and some of my team members are during that period of time are quite busy with traveling.
Okay.
So I decided to skip it this year, but maybe next year. Yeah. That's it.
I've been in contact with Shah quite a lot. Maybe he mentioned that.
How?
With Shah. We're discussing some projects together.
No.
Shah Berchi?
No. I haven't heard anything.
No, so we are a small community, so it's always a like family reunion meeting.
Yes, exactly. You're always there, I guess, in every meeting, right?
Pretty much for us, like it's like I meet so many old friends there, and it's always good chance. Also, it's for me, it's a good deadline. So to keep my students, so you want to go to SED, you finish it. So they have to like finish their abstracts and get their presentations together.
I see.
And as a bonus, they get to go to the conference then, which is a treat, not to say like a.
Yeah, I see that. Yeah. I guess that we are. It's a time to start, I believe. By the way, Shime is also there. I have seen.
Hi.
Shime is sorry, I didn't say hello. How are you today?
Hello, Ham. Hello, Professor Dirks. Nice to see you.
Oh, there you are.
Hi, I'm Simei.
Nice to meet you. Hi. Thanks
nice
for your emails.
Thank you very much for your support.
Oh, thank you for your email and for organizing everything. It's easy for me. I just press a few buttons and everything's organized.
So should we start, you believe?
Yeah, I think we can begin. And I think more students will join the meeting later, gradually.
Okay, absolutely. So I guess that I start. So officially, we are recording the meeting, of course. Information for audience, and hello everyone, and welcome to another online forum of Frontiers in Bionic Engineering, organized by the International Society of Bionic Engineering, ISP. I'm Dr. Hamed Rajabi, a senior lecturer at London South Bank University, and I'm honored to be your chair for today's forum. I'm especially very excited because we have a very special guest today joining us, Professor Jan-Henning Dirks. Professor Dirks has been more than a mentor to me. He has been a guiding light throughout my academic journey for the last 12 years, and it's a privilege to introduce him, as he has been a significant influence on my research career. Professor Jan-Henning Dirks pursued his studies in biology and physics at the University of Bielefeld, where he delved into biomechanical properties of insect antennae. His academic journey continued with a PhD at the University of Cambridge, where he explored the biomechanical principles of adhesion in insects under the supervision of Professor Federle. Following his PhD, he furthered his research on the fracture properties of insect cuticle during his postdoc at Trinity College Dublin with Professor Taylor. Throughout his career, Professor Dirks has made significant contributions to the field, including his work at P&G in Frankfurt, where he focused on competitive intelligence, and his tenure as an independent group leader at the Max Planck Institute for Intelligent Systems in Stuttgart. Currently, he serves as a professor for biological structures and biomimetics at the Biomimetics Innovation Center at Hochschule Bremen. Today, Professor Dirks will share insights into the evolutionary adaptation of skeletal structures in animals, ranging from insects to starfish. His expertise in this area promises to shed light on the fundamental principles and potential biomimetic applications. Professor Dirks, I very much welcome you to our forum.
Well, thank you very, very much, Hamed, for this very kind introduction. It's been a pleasure to work together with you in the last, as I said, just more than 12 years, and it's been amazing to watch your fantastic scientific career and to observe it. So I'm very, very happy that we have spent the last years working together. And thank you very much for inviting me and organizing this great opportunity to talk about biomimetic research here in Germany. So as I said, I'll talk about I picked a few selections of the research that I've done in the last years to highlight, that's the idea of this talk, to highlight a few aspects of fundamental research and how this is connected to biomimetic research, because that's probably also one of the take-home messages which I'd like to reveal, to emphasize today to all of you, the young biomimetic researchers. I'm not young anymore now. To not forget about the fundamental research. So biomimetics is an exciting and fantastic topic, as Hamed certainly will agree on. And I'm doing this every day with my students, but at the same time, you will see hopefully in my research that I'm emphasizing the fundamental aspects. We have to understand something first before we actually can transfer it into technology. And today I'll show you a few selected examples of my work from insects, and from starfish, and to highlight what we've done and the team that I've been lucky to work on. Before I get to start for you, I think very few of you probably have been to Bremen so far, or at least to Germany. So I'm working at the Hochschule Bremen and the City University of Applied Sciences, which is, I'd say, the north of Germany. You can see here, Hamed, probably you're somewhere up here on the left upper corner in the United Kingdom. So this is the state of Bremen, and we are the Hochschule Bremen, so University of Applied Science. Here we're one of Germany's leading universities of applied science, the top 7 regarding research output. We're very proud of that. Which is kind of have to say, it's like uncommon for a University of Applied Science to do so much research, but we're quite happy on doing this. We have about 8,700 students, 178 faculty members here. So we focus a lot on education and teaching. We have an international course on biomimetics here, a bachelor's course, which at some point, but we have excellent students coming from all over Germany and neighboring countries to do their bachelor's specifically in biomimetics here at the Hochschule Bremen. And we're very happy our students go to all places and all over the world, the Ivy League, and someone to London visiting David and so on. We're very happy to export students. As I said, we have a very strong, unusually strong focus on research and transfer at the University of Applied Sciences. We have a lot of historically grown collaborations with maritime and aerospace engineering. Bremen is a place that basically has a strong connection to aerospace engineering in general. Many parts of rockets are built here. We have Airbus. If I just look outside of my window, I see one of the German headquarters of Airbus very close by. Sometimes we can see piles of rockets being moved down the streets. So that's one of our interests. So it's actually not surprising that that's about 20 years ago, give or take 20 years ago, the Biomimetic Innovation Center here at Hochschule Bremen was founded as one of the leading German institutions for biomimetic research per se. There are some biomimetic parts, of course, at some excellent universities and some colleagues working on this, but we are very much dedicated to doing biomimetic research here. We should be about 7 or 6 professors. Currently, we only have 5 here who are dedicated to different parts of biomimetics. So we have people, excellent researchers working on biological materials, reinforced composites using biological materials. We have a focus on animal locomotion, particularly flight, flapping flight, gliding flight, and these things. Many of my colleagues are working on functional surfaces, typical biomimetic topics you've heard of, lotus effect, Salvinia effect. For most of the audience here, probably nothing super exciting new, but we have the facilities to do this here. We also focus on biomechanics and fluid dynamics. So these are with professors who do this here. And my group here is called Biological Structures and Biomimetics. As you can see here, it is a highly interdisciplinary topic. Again, for the audience, probably not a major surprise here, but this is one of the aspects which also convinced me to join the Biomimetic Innovation Center. As a biologist myself, as a zoologist, I could just walk across the corridor and talk to a material scientist. I can walk across the other down the other corridor and talk to a computer specialist, someone an excellent with FEM, with all the kind of things. There's also the pleasure that I had working with Hamed together with someone who has different expertise than yourself, and you can really get some cool things going if you're not focused on your own things, but you can talk to people, meet them, and approach biological problems from a different perspective. My group here, just to give you to focus down, I do a lot of things with insects, as you will see. I'm interested in the cuticle exoskeleton, the crispy part outside, the cuticle parts, which is an amazing functional composite material, versatile material. I'm doing a lot of fundamental research regarding the ultrastructure of cuticle. It's a surprisingly understudied subject. I'm always amazed if I look into some research questions like, no one has ever done that? I mean, yeah, apparently no one has. I'm not complaining, it's great. I could be easily busy for the next probably 2 or 3 careers just studying these fundamental properties. So I'm looking at functional correlation exoskeletons. That's what part of my group is doing this, how do things work. We use numerical simulations, something where I also work together with Hamed, who's an expert on this. Recently, we've developed more like, we tried out playing around with robotics, we designed some bio-inspired vehicles. We are redeveloping joints. I'll talk about this later in a bit. We have a nice project together with Airbus where we're building morphing structures based on starfish. And we've also been working together with biomedical companies where we're transferring principles found in the insect exoskeleton into biomedical applications like prosthesis, orthesis, which at the end of the day has not many things in common. So you can see there's almost an endless source of inspiration in the exoskeleton. Obviously, I'm not doing this on my own. This is basically the current size of my group. The staff members, we have about 12 bachelor students and a few master students. So I'm very happy. I have about 3 PhD students who are currently working on various projects and 1 postdoc who I keep busy and who keep me busy. And it's a great fun to work together with them. So that's basically the current size of my group, which I'm very happy. It's not too large, so which I prefer. So I can be involved in the research project, which is something which when I became a professor, like something I had to learn. Like, of course, you can't be involved in anything in that detail anymore. Unfortunately, you're more busy with writing grant applications and filling out forms. So I'm quite happy with this side of the group, which allows me to keep in touch with research. Now, basically coming into the probably the aspects which you mostly be interested in is the research which I kept me busy for the last almost 20 years now. My focus, as Hamed kindly introduced, is the exoskeleton of insects. That's why I started my research. And I think this is a very versatile structure, because if you think about it, if you look at an insect, any kind of insect that you have, like just a fly, a dragonfly, a locust, a cockroach, whatever you have, this exoskeleton, this is what determines the insect to such a large degree. Everything you see, the morphology, the legs, the wings, the mandibles, the mouth parts, it correlates, it is connected to, of course, the biomechanics. So what is it capable of doing? And it also allows for a certain range of behavior. At the same time, the behavior, the requirements affects what kind of morphology can I have? So how does it allow me, what does it allow me to do? It's all very closely connected. And in the center of this question from my perspective is the exoskeleton. And understanding more about the exoskeleton can offer us a great biological tool. So it advances biological understanding about the most successful group of animals that we have here on our planet. And at the same time, it can provide a fantastic inspiration for biological inspired technology. So I'm not sure about the audience, about your backgrounds, but I guess there'll be plenty of engineers and material scientists here in the room, and they could ask themselves, why on earth is Jan-Henning looking at insects? Well, you could argue actually the other way around, say, why not look at insects? They are pretty much, if you were an alien arriving from a different planet coming to Earth, it is very, very likely that you will have to deal with an arthropod or probably an insect. There's like 1 million species. They're the most abundant or multicellular organism that we have on our planet. Biologically, we consider our mammals, we are just a niche product. Having our skeleton inside is great, we're used to it, but from the biological perspective, it's actually a niche. The majority of organisms on our planet have their skeleton on the outside. And just to illustrate again the vast number of insects, for you as engineers, probably it's easier to imagine, there are estimates that we have about 1 trillion insects, individuals on our planet. 10 to the power of 18, like a proper trillion. That's a bit hard to imagine, but the number, which is, if you just try to imagine every star in our Milky Way, on every star in our Milky Way, you could put an average sized anthill. That's how many insects we have on our planet here. So we could argue there's quite a lot of them. And so this shows that they're evolutionarily quite successful. They've been here for about 400 million years. And as I said, what is amazing is about the huge variety. This exoskeleton that they are built of, it is pretty much the same Bauplan, as we would say in German, so the same concept, the building, the background is all the same. Every insect is built in a very, very similar way regarding the exoskeleton, the caput, the thorax, the abdomen, wings, legs, but it's just different ways of arranging this. So you have like a Lego box where you have a limited number of parts, but they can take many kinds of shapes, and you can attach them together, and they form this amazing exoskeleton structure, which allows them to fly, to dig, to swim even, and so on and so on. And this is something that piqued my interest. And again, narrowing it down, what I think is really fascinating about the exoskeleton as a structure is that it is actually it's built from very simple biological materials, to argue that way, from fundamental components. We have chitin, which is a kind of sugar in a way. The chemists in the audience might forgive me for saying that, but it's very related to cellulose, and it's one of the second most common biological materials we have on our planet. So with chitin, you have some proteins, and we have different hierarchical levels in which these structures are arranged, and they can form an amazing abundance of material with different properties. Like imagine building an entire airplane out of one single type of material. The transparent windows, the tires, maybe even the wings, the machine, the turbines, and the same in the insect. Everything you see on this picture, from the antenna to the transparent eyes, to the adhesive feet, to the mandibles, mouth parts, to the wings, is made from this cuticle. So it's a very, very versatile material. It's actually hard to say the cuticle because it's like saying plastic. It's a huge variety of mechanical properties. And this has piqued my interest for many years. And so I've selected a few examples of just to highlight or to illustrate what we've been working on the past, say, almost more than 14 years now. I started my work at the University of Bielefeld, and I was working on a robotics group, so to say, more or less. They were building bio-inspired robots. And what they had the need of basically an antenna for active tactile exploration. For those of you working in robotics and in sensor systems, cameras are cool and great, and cameras in a good way, but sometimes you need to touch something. If you're interested, for example, in the mechanical properties, or can this carry is this load bearing? Can I put my weight on something, for example? Or you're in a surrounding where maybe your vision is inhibited or dark. Actually touching something is still a valid sensor system and an important sensor system. And insects do this quite a lot, and particularly nocturnal insects. So we're studying the long, slender antenna of this Causus morosus, the Indian stick insect, as you can see here, and trying to develop like a bio-inspired solution. Because if you think it's this long, slender antenna, it can easily bend, but it should not break. And if you let it go, it moves backwards and it shouldn't oscillate. So it actually has quite a high number of mechanical demands, which are important for such a thin, slim antenna. And we analyzed the ultrastructure, the morphology of the antenna, and we just found that it's basically consisting of this outer exoskeleton. But the secret, or the interesting part, is this conal shape at the base of the antenna, which is a critically overdamped system. So it's endocuticle, it contains a lot of water, and it's like a proper damping system. So the way you can use this antenna, you can still move it. It's a highly controllable system, but if it's deflected and swings back, it's critically overdamped. So it basically does not oscillate. And if you dry it experimentally, it nicely oscillates at 440 hertz, so you can tune your guitar with it if you ever need to. But it just shows the importance of this material, the material properties, and the effect of damping in a model. Carrying on from this, I was still fascinated with insect adhesion, and I picked some examples of my work that as a PhD student, again, many years ago, but this is still an ongoing, fascinating topic, which I still like talking about because it's still a kind of unsolved. So if you look around you, you see insects crawling up and down, maybe in the air condition if you're unlucky, and in your refrigerator if you're super unlucky. And one of the amazing things is that insects can stick to almost all kinds of surfaces. And this has been known, well, for hundreds of years to humans, and insect pests climbing everywhere. And for more than 200 years, scientists have been amazed by this ability to stick to pretty much everything. And we still actually really don't know how this works. 2024, we can send helicopters to Mars, or do all kinds of technological stuff. We have artificial intelligence around the corner, but how do actually insects stick to a surface? So I was investigating this during my PhD with Professor Federer, and it was known that basically insects secrete an adhesive liquid. So maybe you've known this, heard this before, but all insects have sweaty feet. Like, this is a knowledge you have to live with from now on, that whenever insect, they have like liquid sweaty feet, and they have this adhesive liquid. It's not a glue. It cannot be a glue, because otherwise you would only basically be able to play 6 steps in your lifetime. So it has very special properties. We found out it has very likely to have non-Newtonian properties. So it's a shear thinning liquid that the insects can generate friction, but they can also generate adhesion with a liquid, which is not common. But understanding this, so that was the fundamental question that we have, allowed us, and understanding more about how insects feed allowed us to basically trick insects' feet. And it's coming to the biomimetic application, which is not really biomimetics in the strictest sense, but knowing about how biology works, understanding the principle, we could design something or think about it, something that allowed us to trick insects. And we coated, for example, a rod. We found that a special type of polymer has a certain ability that selectively removes water from the adhesive liquid, like a small sponge. And if you coat a small rod here, see a cockroach climbing up and down on Teflon, no problem whatsoever. And if you coat a small rod with this polymer, then suddenly you can see the cockroach, although it really likes to have the apple up there, can't reach the top anymore. So they slip down, they fall down. So it's basically just tricking the adhesive system of the insect. It's non-toxic, basically can last for a very, very long time, but it works, and understanding how insect feed works and then designing something that basically interacts with us. Again, the problem was it works too long. So industrial applications are very limited because the company can only sell it once. At the same time, a project I did together with Stas Korb, many of you probably know him here, at Kiel. So we also looked at this critical roughness. So insect adhesive systems, they are good at sticking at smooth surfaces, at glass, for example, because they have this adhesive liquid, we call it that, and they can make capillary adhesion together with friction and generate friction. But they also have their claws. The claws play an important role to attach to certain surfaces, so if they are rough, for example. And what is nice, there's basically a kind of Achilles first. So there's a very dangerous roughness for an insect. If it's very rough, they can use their claws. If it's very smooth, they use their adhesive pads, and they can make close contact. But there's like a death zone of like, this roughness it's too rough to use the adhesive force, the adhesive organs, and not rough enough in some way to properly use the claws. And if you can design a surface that is exactly in this range of roughness, then we developed a method basically to test this. So you can see here some cockroaches and some other insects who climb up, and you see this slightly shaded area here is covered by this micro rough patterning, and they can slip. It's not 100% proof of, but it's non-toxic. It can last for a very long time. It's basically a micro structure with 2 wavelengths that is embossed into a plastic film. And you can see the insects, they don't really like it. They try what's going on here. This is slippery, so it prevents insects from climbing across certain barriers. So there's something that we also, but again, we were only basically able to design this, and Stas Korb also in this group were able to design this because they knew, and we knew, how insect feet actually work. And you can do some maths and engineering and say, okay, if that's how it's supposed to work, this are things that basically should. Carrying on from this this work on insect adhesion, I then moved on to studying the fundamental mechanical properties of cuticle. As a biologist by training, I thought, okay, I'm interested in biomimetic things as well, and most of them, as you are probably here, but I needed to learn proper engineering, to put it that way. As a biologist, you don't get really good training in mechanical engineering and fracture toughness, or in what is a Young's modulus and an A module and all these things. So I decided to do my postdoc in the Department of Mechanical Engineering in Trinity College Dublin. I had the great pleasure of working together with David Taylor, who's a world-known expert on fracture mechanics. And we started to investigate the mechanical properties of insect cuticle, really starting from scratch. Say, okay, now as a biologist, I have an idea, I could do some tests, but every engineer looking at my tests will say, well, this doesn't make any sense from the engineering perspective. You're using the wrong tools, you're using the wrong measurements, you should do it properly. Vice versa, some engineers studying biological materials, they sometimes do things where as a biologist you say, oh no, this is not good. The material is dried out, it's far from the biological organism. So you have the right tool and you do the right measurements, but it has nothing to do with biology anymore. So what we did, David and I pretty much brought it together. He's an expert engineer and I was a biologist with some training, and we brought it together. We tried to study cuticle in its natural system. And we first of all started with measuring fracture toughness. We were lucky that we were the first ones to actually determine the fracture toughness of the tibia. There was a value that was not known so far. We placed incisions into the legs and so on. And we found that a cuticle has an extremely high fracture toughness, meaning like resistance to crack propagation. It's very good in resisting crack propagation. It's very difficult to drive a crack through many parts of the insect exoskeleton. And we then basically thought, okay, well, let's have a look at the wings. And this is, I guess, where Hamed caught our interest. And we thought, oh, some other crazy people are working on insect wings. So what David and I did is where we wanted to understand maybe the wings are even more fracture resistant than the tibia, again, chasing for great material properties. So we took a hind wing of a locust. You'll see this in the video in a second. We placed a small incision into the wing. And then we started pulling on it to see what happens. And as you can see, at some point, the crack here, the notch will start to grow, and then basically it will hit a cross vein, as we call these veins. And the engineers in the audience will see that there's something interesting happens. It's delayed, so it's stopped by this cross vein. And then at some point, the cross vein breaks. The machine is luckily strong enough, it was very expensive, to break an insect wing. So it runs through the entire wing, and then it propagates even further after breaking the next cross vein, and so on and so on. And this very simple experiment allowed us to measure certain mechanical characteristics of the veins, of the membrane itself. We also were able to determine like an optimum spacing between the veins, which is the maximum fracture resistance of the structure at minimum weight. And we're very excited by this. And oh wow, we can patent and all these things for biomimetic, but nah, not really. The people in engineering had found this ripstop technology for quite some time in parachutes. Biology has found it a few 100 million years ago, but we were quite happy that we could understand how this whole thing worked. And we started for some really nice collaborations together with Hamed and his team, doing some exciting simulations on stress distributions in the veins and in the membrane, as things that we as biologists were never able to do. We needed engineers to help us with this. So that's, again, some fundamental things that we require to understand how does cuticle work. And from this on, some projects, fast forward a few years, that Hamed we just talked about at the beginning, my centrifuge experiments. As a biologist working on the cuticle for such a long time, there was a major question that I was very much puzzled by, is the ability to react to mechanical load. So your bones, you're all vertebrates like I am, and you have bones inside of your body where they should supposed to be. And one ability of pretty much every biological material that we know of, particularly wood and bone, like skeletal materials, put it that way, or load-bearing materials, is their ability to react to mechanical load. If you're an astronaut going into outer space, one of the major challenges, or like medical challenges, is the loss of bone material, because there's no stress. The skeletons don't experience any stress anymore, and your body says, well, I don't need the material, I'm going to basically reabsorb the bone and do decreasing bone density because it's not required. It's like weight saving. Same problem, osteoporosis. With this balance of building bone, removing bone is not working properly anymore. Your body reabsorbs more bone than it actually builds, and Some people might experience osteoporosis at some point. So bone has the ability to react to mechanical load. That's lots of things are known about this because it's medical material. Same is about wood. There's compression wood, there's tension wood. If you talk to someone who's working in forestry or someone who has a proper job working in building his furniture or with wood, not an academic like us, they will certainly tell you that there are different types of wood, and wood reacts to the forces it experiences during growth. So it can be a wood, a tree growing in constant stress by wind will grow different types of wood and so on to optimize the use of material. So we know about wood, we knew about cuticle, and something that puzzled me a lot. What about cuticle? So I'm a great fan of cuticle anyway, but it's the second most common biological material in the world. And we had no idea whether cuticle is capable of this fundamental aspect. So whether it's special, does it have it? Does it not? We don't know. Or whether it's basically this ability to react to mechanical load has been an evolutionary development of bone and maybe an evolutionary development of wood, which are close or wide apart. So where does cuticle stand? And my PhD student, Karen, and I'm very happy that she was well trained by Hamed and his team. She took over this project some time ago and did a PhD. And we built a large-scale centrifuge. So the idea is we had large cages with these insects, with the Locusta migratoria, as you can see up here, Chistocerta gregaria, and they were growing up at high gravity conditions. So this cage, you can see here in this cage, they had like some place to hide, so that's like as good as an insect can live in a cage, and fresh food, everything they wanted, and they were subject to increased mechanical load for several weeks. We could go up to 10 times the gravitational load, the maximum, but I'll show you in a few slides that was not very well received. But we could easily go to 3G, 5G, and 8G for a continuous period of time. It's like these insects getting up in the morning and weighing 3, 5, or 8 times their body weight, still being able to move freely in theory and grow. So when an insect is put into the centrifuge, there's about a time frame of 2 to 3 weeks in which it deposits new cuticle during the maturation time. And we used this period of time, so the insects hatched to the final form, to the adult insect, then they were placed into the centrifuge and lived there for 2 to 3 weeks. And at the same time, obviously, we had the control experiments. We then took their tibia, as you can see here, this is our standard biomechanical body part, because again, coming from the engineering perspective, it's almost like a tube. It's not a perfect tube, it has some deviation, which are also meaningful. But for a first approximation, it can be considered as a tube to some extent. And then we performed some kind of standardized cantilever tests, which allowed us to then measure this maximum strength and stiffness. So standard engineering parameters. And then we compared reference or locusts locusts raised under increased mechanical load. As every engineer will know, obviously, we need to have some morphological data on the locust as well. So first, we had to develop some new methods to actually stain and to measure the complex morphology of the tibia hind leg. So we use micro-CT technology, which is very powerful tool to get 3D information on complex biological materials. So here we developed a method which can help us to a semi-automated differentiation. We need to know which is the crispy part, the executed, and which is the soft part. And it's very tedious and time-consuming to do this in the standard way of cutting and so on. So Karen, my PhD student, she developed a method, the staining method, which then can be used in combination with some clever image algorithm methods to then stain or scan part of the cuticle of the exoskeleton and get proper 3D distribution of different parts of the skeletonization of the cuticle. We use this data, and then we're able to show that actually, yes, something does happen. So an insect at control conditions, 1G here has a typical shape of the leg. I'm not going into too much detail here, but insects raised at higher gravity conditions have a different type of cuticle. The cuticle deposit has different mechanical properties. We corrected this for starvation and for body weight and body mass and all these things. So there is something happening. This was our major finding, that we don't exactly know what is happening. So in terms of, we have to go to the microscopy level and see what's really happening with the orientation of cuticle and so on. But we are very certain that something is happening. We also saw this when we looked at the Young's modulus here, just as a selection of this, that at an increased stress to 3G, actually the legs become stiffer. the Young's modulus increases. And if you increase the load even more, 5G, 8G, then other effects kick in, which we are not entirely what happens, but then they go basically back to the normal. We presume there could be some effects regarding fitness, starvation, anything, but at least if you are an insect that has been raised at 3 times your body weight, like you going to the gym, lifting your body weight every day a few times, this increases the actual mechanical Young's modulus of your cuticle. So we're very excited to get this published, because it shows us that yes, the second most common biological material in the world also has the ability to react to mechanical load. This is something we can say about 80% of the world's species presumably has this ability. So this, again, will kind of keep me busy for the next 100 or 200 years of research to figure out what's actually happening here. But it's like a go, no go thing. We were very excited to get to see this happening, and many open questions will follow up this. Another more recent project, which I'd like to show you, just to illustrate what's happening with the exoskeleton, is maybe you've observed that some insects have these exciting, the jumping insects, the locusts have this interesting ridge-like structure on their femur. And I asked myself, okay, interesting skeletal system. And one of my very talented bachelor student, Lara, she investigated this ridge-like structure. We asked ourselves, what's the advantage of this ridge-like structure? Because it's a very characteristic feature. And she did some micro-CT studies. We found a very nice correlation of the muscle fiber attachment to this ridge-like structure. So it's a mechanical reinforcement. You could ask yourself, why is it not smooth? Like, why do we have this Christmas tree pattern or fish bone pattern, as we call it? And she did lots of interesting micro-CT studies investigating this, and then she did the actual biomimetic thing. And she looked at it, okay, what's the fundamental principle behind this, like a selective reinforcement with a certain pattern. And what we found is that this, again, for most of the engineers, it's not a major super surprise here, but I'd be happy to see this in the insect represented, like a more evenly distributed stress if we have an arched structure and reduced overall stresses. So it's a good structure that presumably helps the locust to evenly distribute the load of this massive jumping muscle, which is inside of the femur. So again, it's no decorative element, but it has like a functional system. For biomimetics, you could ask yourself, okay, most engineers would say, oh, that's not a surprise, I would build the structure the same way. But for us as biologists, it's interesting to see that biology has found the same solution a few 100 million years ago. Same with the wings. Coming now, basically, we talked a lot about insects. I'd like to show you, as promised, a few other aspects of my research which don't have too much to do with insects. So moving into different types of skeletons, which are technically not real exoskeletons like insects have, but they behave in some way as a functional exoskeleton. So your skull, for example, protecting your brain. It is biologically, it's an endoskeleton. It's bone, it's a mesodermal endoskeleton, but functionally, it's a sphere with a high compressive strength, which protects your brain. Your rib cage, same thing. Technically, like biologically, it's an endoskeleton, your ribs. Mechanically, it's more an exoskeleton because what it protects is inside. So there's sometimes in biology, this separation between exo and endoskeleton is not entirely clear. Looking at a snake or a bird, and lots of interesting exoskeletal features that we have. When we started looking at this, as a small video I'd like to show to you. We developed an underwater robot based on some soft exoskeleton. We thought, well, why do exoskeletons always have to be stiff? We had also a very talented student who just finished his PhD in David's group, and this was a specialist student project which just took 9 weeks, which is mind-blowing. He's extremely talented, and we developed like an underwater robot, which is inspired by the kind of the outer shaping of single cellular organisms. If you think about single-cellular organisms, for example, like single cells, they have usually very soft. So they run into things and they deform. So you try to run into things and not deform. Most of like car engineers would do something else, but your body should stay rigid. If you hit your head, it should protect everything that's inside because it needs to be protected and it's very rigid and stiff. Bacteria, or even like single-cell organisms, they have a different approach. They just run into things, but they don't care. They deform and they regain their mechanical shape. They are They have a structure which allows them to deform, and this has the implication that you need less technical hardware to prevent collisions. So yeah, I'm going to collide. I don't care. I don't damage myself. I don't damage you. I can bump into things. So that's the idea that we just tried out here. And I like to share this video. I hope you can see that. Can you hear the sound?
We can hear, but not very loud.
Oh, sorry.
On impact, the rigid encasing poses a threat to the fragile surroundings, such as coral reefs, shipwrecks, or accompanying divers. Nature has dealt with this issue since the very beginning, which is why we can find a solution to this problem in the simplest of life forms. Most unicellular organisms, such as paramecia, have a rather limited sensory input and therefore collide with each other and their environment regularly. But instead of having a rigid shell, they are encased in an elastic membrane. The same principle can be applied in robotics. A soft exoskeleton, inspired by the flexible outer membrane of unicellular organisms, is a versatile solution and eliminates the need for an infallible navigation. To verify its effectiveness, we have built a fully functional prototype equipped with such an exoskeleton.
直播声音太响，那个什么仿生协会那。
Maneuverability and 6 degrees of freedom. To control them efficiently, we have developed a simple neural network based on interlinking PID controllers optimized through an evolutionary algorithm. This enables the system to quickly adapt to changing environments and load distributions whenever additional equipment is attached. In our prototype, the soft exoskeleton is entirely 3D printed using a thermoplastic polyurethane. Contributing to the spherical design, the thruster ducts are elongated and created from the same material. As the spacing allows for multiple cameras to be attached, the exoskeleton prevents impacts from causing damage in any direction without limiting the camera's field of view. Our experiments have shown that the installed soft exoskeleton significantly decreases the stress on all components by measuring the net acceleration during multiple impact trials. Even when operating at comparably low velocities, the exerted forces resulting from an impact are reduced by 50%. As the exoskeleton does not affect the maneuverability of the system and has a negligible effect on its buoyancy and maximum velocity, it has proven to be a versatile solution for any type of underwater application. We have already run first trials with the SAUV in flooded caves and plan to develop its autonomy further to be able to use it in other complex environments, such as shipwrecks or coral reefs. The soft exoskeleton is a highly effective yet low-cost addition to any newly developed or existing system to make them safer and increase their longevity, because once again nature has taught us it is better to recover from an inconvenience than to expect to never encounter one.
Yeah, can you see here? It's like basically a bumper. Again, you could argue, whoa, okay, yeah, it's a technical system which has been running for some time, but attaching these to underwater vehicles, we're surprised just to get this excellent feedback on this, and people are very interested in using this technology underwater as well. So from the single-cell organisms, as promised at the beginning of the talk, coming to starfish, something which I started some years ago as a side project and now developed into a full-blown academic research project with more and more people associated. It's a collaboration we have together with Airbus here in Europe, and the idea was that the technical need for morphing structures is surprisingly high. Morphing structures is a structure which can change its shape. In biology, it's very, very common to be able to do that, but in technical systems, it is actually not that straightforward. Everyone has ever used an airplane. If you just look outside of the window, the wing has so many technical features in it to mimic a morphing structure. It changes basically its shape during the flight. When you take off and you land, the flaps go up and down, which is basically changing the aerodynamics. But you need like lots of technical hardware to get this to actually work. So aerospace engineering is very much interested. They've been researching on that for many, many years, trying to get morphing structures in airplanes or in aerospace engineering in general. Change the shape of your wing, change parts of the airplane to make them more versatile. At the same time, it has to be failure resistant. So if the electronics fail, you can't just have your wings falling down, which is unfortunate for an airplane, obviously. So far, engineers have been using classic mechanical joints to manufacture morphing structures, which is a major challenge. They weigh a lot. They have to have certain parameters to fulfill. They are usually not per design fail-safe. For example, your body is as a vertebrate is built of ball and socket joints to a large extent. If you collapse, if you become unconscious, you collapse. You just drop on your desk, or worst case somewhere else, because you're missing your muscles. Your muscles keep your body upright, and you constantly use your muscles when you go for a sailing trip, or then why are you tired in the evening? Because your body is constantly using muscles to keep it upright, to stay where you are. There's one application, aerospace engineering, but if you look around your office, your house, there are countless applications for morphing structures. We say, why not replace this joint, this mechanical classic joint, which has been known for hundreds of years, with a more elegant morphing structure, with something that can change its shape without the need of joints. For many engineers, this is hard to imagine because we're so used to joints, but joints are not necessarily always required for a kinematic chain. And this brought me basically to my research as a biologist, giving some lectures on starfish for my first year students. I encountered something which puzzled me, something I didn't know firsthand, is that starfish are echinoderms. You see their skeleton in a second. They have the ability to hold their skeleton upright on a certain position without the use of energy. So if you were to basically, a starfish can reach out his arm and then interlock the bones it has inside with a special tissue that's surrounding these bones. You use your muscles. So if you, for example, were to hold something in your hand and I ask you to keep this for 10 minutes, 15 minutes, I guess very, very few people will be able to hold it that long. And at some point, because of your physiological aspects, your muscles will fail. You can't do that, even if you have muscles like this. Starfish can do this because they have a trick. They interlock their skeleton using this mutable collagenous tissue. And we then basically said, okay, we want to understand how does that work and transfer this into engineering. And one of the first things we did is we scanned starfish using some high-resolution XRM scanning technology. And for us, the exciting part was also, we were one of the first to actually be able to scan an entire starfish at a very high resolution using a special kind of micro CT. So not only scan like a small part of it, which was known before, but really to investigate and to understand how does the skeleton really look like, and as a biologist to see how does these thousands of bones interact. We have way more videos on this, but this just gives you an idea that this whole entire endoskeleton of the starfish is built of thousands of small interlocking ossicles. That's what they are called. And the first thing, again, what we want to know is, how do these ossicles work? Like they know that they're ossicles and they are very porous. I'll show you pictures in a few seconds. And the very first question that we want to answer is, coming from our cuticle inspiration, are they always the same? Or are ossicles also able to basically change their porosity and their shape? So you can see here my workgroup, we're almost working through the entire cast of SpongeBob here. We have basically we knew that exoskeletons from arthropods can react to load. We know that bones can react, and we want to know, is the same true for starfish ossicle? And this is like one ossicle that we scanned at a very high resolution. It's like one of these hundreds of thousands of bones that is present in the skeleton of the starfish, and you can see it's a very porous structure. And first of all, we were amazed by this and very happy. And we thought, okay, this porous structure is getting very similar to the porosity we find in our bone. Evolutionary, they are our ancestors to some extent, right? Not really, but they are like an evolutionary super old bone structure. We as vertebrates, your bones are evolutionary much younger. They're like hundreds of millions of years of more evolution. So this, we consider that we can look into our past, evolutionary past, and we already see a porous structure. That was very amazing. And we were the first ones to actually be able to get the full 3D information of the porosity of the entire ossicles. We have thousands of scans of these. And coming in the engineering part, we want to analyze the porosity and see how does this work, how do ossicles work. And the nice thing about the starfish ray is the arm of the starfish here is we have a kind of time capsule. So when a starfish grows its arm, when they become bigger, they always add new ossicles at the top of the tip of their arm. And the older ones just grow bigger and bigger and bigger. So if you look at the base of a starfish arm, you see an old ossicle. And at this tip, you see the same type of ossicle, but just much, much, much younger, like a baby ossicle. And here you see the adult one. And you see a teenager in the middle. So by analyzing the porosity of the ossicles at different ontogenetic stages, developmental stages, it's like taking your body and having a young femur, and a medium femur, and an old femur. And each of these femurs, ossicles, experiences different loads during the time of the growth and then during the. So we were able to see, does this correlate with some kind of mechanical load? And we actually saw that yes, the porosity changed. I'm not going to go into too much detail here, but the porosity of these ossicles, the overall porosity changes. So it depends whether you're a base, middle, or tip ossicle, your porosity changes. So there's some correlation of mechanical stress and the porosity that's built by the starfish. We then looked at them the mechanical stress that's experienced by the ossicles. So this is a group of ossicles, you see the video here, and we were also able to visualize the muscles. So this is colored in red here. So the muscles which connect these ossicles, and we have a very good knowledge now where these muscles attach to the ossicles, where they pull, and where tensile stress is basically applied to these single ossicles. And then we used FEM, again, as Hamed's favorite tool for many things, problem solver and problem generator for many stuff, things that we cannot do biologically. And we performed some, again, to simplify it down here, we performed some measurements of where we were able to show that parts of the ossicle that experiences muscle-induced tensile stress correlates very well with different types of porosity. So again, this just might not sound super exciting for the engineers, but for the biologists, this is really exciting because we can show that even our ancestor bones, technically, already have the ability of a correlation of bone structure and mechanical law. They don't just build something, it's built basically according to the mechanical need. We then looked at muscle attachment site. We were able, this is very nice, you can see here on the pairs left and right, there's always muscle attachment site and free part. And we can again show that where muscles are attached, there's a different ossicle porosity and a different shape of the porosity, which then more evenly distributes the load that the ossicles basically experience. Something we also found then in the compressive parts. So when ossicles touch each other, there are some occasions on the side basically of the ray where ossicle is touching ossicle, really like exerting mechanical compressive force. And then you can see here in these images, it's magnified here, that wherever this left is one ossicle, on the right is another ossicle, and wherever they touch, again, there's a reshaping or the reformation, or at least a different part of ossicle porosity that is formed. So the ossicle basically builds the corresponding porosity that's required to deal with compressive stress. We then analyzed, is actually an ossicle reshaped? So do they reabsorb material, or can they only build material? And just looking at the time, shortening down here, it looks that they only can basically build material. So unlike us, our bones can build and remove material, and it's what we presumably found in our data indicates that they only build new material. So in the core as shown here, if you look at an old ossicle, there is a baby ossicle still inside of that old ossicle. They're not rebuilding the entire ossicle when they grow, they just add layers and layers and layers of modified porosity on top of the old ossicle, and they don't modify the inner part. In your bone, it's different. Like your bone constantly, entirely remodels inside, outside, and ossicles don't do this. From this, we developed, I can't talk about too many details here, but we developed a morphing structure, which is based on the principle of the starfish. This is one of the older prototypes. Again, I apologize, like, I'm not able to share the newest developments that we have here. But again, the structure can adapt to many types of loads. It has a local controllable stiffness, many degrees of freedom. We have a patent which has just been granted. We're very happy about this, which is a surprisingly simple solution, and we were surprised that no one ever thought about that. It's a very, very powerful tool. Airbus is very much interested in applying this. We now brought it to the point where we can also move it in some way. You can see here this like a master's project that we can actually move the structure, so we can make it to deform, and then we can apply biomimetics, for example, very simple tools to actually get it to move in a controlled way. Another master's student of mine has recently finished. We also looked at the starfish again, and how can we scale this up, right? There's no point in just being able to build something very small if you want to cover an entire airplane or build an entire table or something like this from this the morphing structure. So we developed some tessellation aspects where we are able to scale it up in certain ways to build a morphing structure that can cover a large area and that can has a specialized functionality. And so at the end of the day, when we have several this is like older prototype. This is a way where we basically can form all kinds of structures. And the nice thing is it doesn't require energy to stay in this shape. It can be easily changed. It can self-heal at the same time. And we have basically unlimited applications. We're currently collecting all kinds of application ideas with companies to use this. With a phone holder, obviously everyone needs a phone holder, or if you have a pen, or like if you have to hold your plant. There are way more sophisticated things where you can use this, but we're very happy that we found this quite elegant solution to this. So for you, take home message as the time is running out for you. First of all, I hopefully been able to convince you that insects are great. And starfish are also pretty cool. All insects have sweaty feet. Don't forget that. Next time a fly is crawling across your window, it will have sweaty feet. I've shown you that it's quite new thing that we found that insect cuticle can react to mechanical stress, which doesn't sound that amazing to you maybe, but it was quite interesting for us biologists. You maybe for the first time in your life have seen the inside of a starfish skeleton to some extent, and maybe you learned that starfish have thousands of small ossicles, and they have a surprisingly high number of similarities to your bones. So your cousins are of starfish, they actually have something quite in common. As you see them in the examples that I've shown you, I've tried to point this out, and I hope I got the message across that biomimetics is a fantastic tool, and it drives our research, and it gives us the questions pretty much that we need to answer, technological questions. But I will try to emphasize here that we very often need to take a step back and do some fundamental research first to really understand what's going on, because then we can make a much better decision whether it's worth pursuing this, or we actually don't just copy something from biology, but we actually transfer the principle into technology, because then it's way more powerful. It's way more groundbreaking, to put it some. You can rethink things to some extent if you really understand the principle, not say, oh, it looks like a starfish, I'm going to build a starfish. But you have to understand the idea of what's behind it. And this is something which I'd like to encourage you as young biomimetic researchers to do biomimetics. It's a fantastic field, but don't be afraid of taking the step back and thinking, what am I actually doing here? And is this like, do I really understand what's going on, or am I just copying something because it looks similar to biology? Last minute, some shameless advertisement here. We're currently starting a new core facility here at the Hochschule Bremen. So everyone, we have it's funded by the DFG, which is very generous. We're getting a state-of-the-art environmental electron scanning facility here. For those of you who know scanning electron microscopy, that's really cool. But environmental scanning means we can do this in almost no vacuum. So in environmental conditions, there's no need to coat samples, no need to prepare them. You can take biological samples, biological materials, test them at high magnification, at electron microscopy magnification, measure fraction properties, measure mechanical properties, observe how cracks propagate. So if you have the need of all these things, feel free to get in contact. We're very eager to try out stuff. Right? So we have you have a brand new machine, you want to see what it can do. So if you have the need, please feel free to contact me. With this, I'd like to finish here. The number of people I could count, I was thankful, is probably endless, including Hamed, obviously, for his inspiration and his ideas. These are just the people who are involved directly in the research I've showed you here. As I said, it's an interdisciplinary group. We have people from all our the disciplines, mechanical engineers, material scientists. Also, if you are interested in joining or coming around for an internship, or please feel free to contact me. We're very happy to discuss any options. So thank you very much again for the invitation, Hamed, and I'm very happy to take any questions.
Thank you very much, Jan-Henning, for the very beautiful talk, very inspiring. Of course, we are open to questions, and everyone is welcome to ask the questions. Maybe before someone asks the question, I jump in and ask the first one. So Jan-Henning, you mentioned about your recent study on the changes in the cuticle properties when the cuticle is subjected to increased loading. I know that you mentioned we don't know much about things that happen there, but you have the micro-CT scans. What do you see exactly there? I see changes in the thickness quickly after in the slides that you show, But is there also any change in the density? I know that we can't talk much about the microstructure of cuticle because that needs other techniques, right? But do you see anything, that can be that is obvious? For example, changes in the density of the cuticle?
Not at all, no. So
Not
it's an excellent question. So with the micro-CT, you can see the X-ray density, of course, right? So you don't really see the sclerotization that much. The sclerotization has almost no effect on the X-ray density. So from our perspective, we didn't see anything obvious regarding the density in our X-ray. So therefore, the follow-up projects, they're already written up. They're just we're waiting for feedback. They will involve like proper biological histology, where you really look at with my light microscopy, and you see, do we have an orientation of the chitin fibers? Do we have different amounts of chitin in various types of cuticle? This is actually this is a super fundamental question which we have to address next. Absolutely, but we can't see this in the X-ray images.
I see. Yes. Okay, that's very interesting. Okay, so but of course this is something that you would like to study more, right? Because you mentioned that it is an ongoing.
Yes, exactly. So that as I mentioned, we with our project, with Karen's project, we've opened up the door. Basically, we said, okay, there's something happening, and now we need to like, when is it happening? What age is it happening in all insects? We have just verified this again. Is there like a time frame in which this is happening? Is just continuous load? Does it have to, are peaks of load enough? It's like you going to the gym, building up muscles. Like some people say you have this interval training. So I say it's enough to do this like high interval for 5 minutes. Others say no, you have to run for 5 hours. So kind of similar experiments we would have to do on the insects to see, is there like a certain window of opportunity in which the insect experiences stresses and then it reacts, or is it continuous application of stress? Can it rebuild? So if you take it out of the centrifuge and wait, does it regain?
Mm-hmm.
It's as I said, I could spend the next 200 years with many scientific careers answering these questions. For Karen, it was a 3-year project with a PhD, and it was very time consuming because we had to develop most of the protocols. We had to be very certain that the sample numbers are surprisingly high. We test like, we've seen it, it's like 200 sample measurements per data point, because the differences are small, but they're reliable. That's something that we wanted to really know. So therefore, we are busy for the next years.
Yeah, okay, I see that. Thank you. Again, I can see the list of all audience. So if anyone has a question, please jump in and ask your question. Maybe I ask another question here, because along the same direction, because you very beautifully communicated the fact that whole body of the insects is made of cuticle, and of course this having cuticle in different body parts has different requirements, right? So this cuticle, the properties of cuticle should change from one to another body part. And I believe that you have published a paper, I guess a review article, how the cuticle property changes from one part to another part. But if you're talking about changes in cuticle properties, of course, I can imagine that, for example, density could be a factor that influences these changes in cuticle properties. Microstructure could be another factor.
Absolutely.
What are these properties that can change cuticle properties, let's say from a very rigid body part, like the elytra of the insect, very protective part of the body, to a very flexible part like a joint?
Yeah, absolutely. So that's the exciting thing. So if we talk about the cuticle, it's actually, it's not a very good way of saying it. Yeah, that's a good point you mentioned in this paper that we've published on this, where we showed there a huge variety of possible body parts, because most engineers, and I'll just pick out something and say, oh, look, I found something about cuticle. And it's, again, it's randomly picking a part of your car and testing and saying, oh, my car has a Young's modulus of 3. It's like, no, certainly not. So the mechanical properties regarding the Young's modulus, the maximum tensile strength, compressive strength, you name it, everything you can think of, fracture toughness, particularly the Kc value, are very likely to vary extreme between the different body parts, between the protective part, the elytra, the wings, which you are an expert on, the membrane, which obviously has some interesting properties, and again, different composition, a very different composition to the rest of the cuticle to some extent regarding chitin resident proteins. So what we think is that the joints are very interesting, the protective parts are very interesting, and we will certainly see differences in Young's modulus in maximum strength and fracture toughness between all these body parts. But again, this makes it also difficult to draw a scientific conclusion, right? So we always have to ask ourselves, what do we actually want? I can spend hundreds of years with countless students characterizing every body part of 1 million species. What's the point of that, right? So what we are mostly interested in is understand what the question that you also brought up is what determines these mechanical properties. So at the end, I'll say, okay, if I know this chitin orientation, if I know it has this much residence, if I know it has these proteins, and I have this amount of cross-linking and this water content, it will most likely have this Young's modulus, or it will have this maximum compressive strength. This is the holy grail of basically cuticle research. So what we are currently doing, because it's as you know, it's a very small community, we are collecting data points. As my group, we're always trying to stick to a limited number of insect species and investigate them as thoroughly as we can. I could easily go to the next forest and pick up another 20 species and publish 5,000 values about Young's modulus, but they're not super exciting. So we are trying to, as I said, to understand the ultrastructure and the functional correlation between this ultrastructure and what we see, the actually biologically relevant exoskeletal structure. And there we are, because composite structures are a nightmare probably to predict and to model, as you probably know. Therefore, it's very difficult and time-consuming and challenging. But we don't give up. We're on it.
Yeah, and if we go back to the fact, I guess this paper of Julie and Vincent saying that the cuticle properties could change over, let's say, seven orders of magnitude from one to another body part in the same or different insects. What does your results show? You know, I mean, the results of your investigation into the literature. Is this very much true? Is it the result of the use of different method or methodology, different type of measurements that we have done on insect cuticle, let's say using a different technique to measure the, let's say, elastic modulus of cuticle, or is this a real fact?
Yeah, it is certainly a real fact that we have a huge variation in mechanical properties. You can easily see this by just touching a single insect, looking at the membrane in a leg and say like the mandibles. They have orders of magnitude of different properties. So I fully agree with what Julian has published in the review, that it covers orders of magnitude. The challenges or the danger is that we talk about probably the wrong way. We have to be aware that people have tested various kinds of cuticles and various kinds of insects and have sometimes used different types of mechanical tests. So it's super difficult to compare in between studies, which is something which we try to make people aware of, particularly engineers and biologists. Engineers are more aware of that. They would say, oh, you can't easily compare nanoindentation with the cantilever test, right? You will get values, you will get both values for both methods and argue which one is the true value. You can argue, oh, nanoindentation is better, or I can say cantilever is better. That's a scientific argument. But engineers are aware of that. Biologists will say, oh, look, I found this paper, and they present a Young's modulus of 3 gigapascal, and in that paper they showed 5, and so it's the same. So, well, yeah, but you should know that they use different methods and so on. So the awareness that is of the challenge of this interdisciplinary exchange is that we as biologists have to understand exactly the question that you posed. Different methods will provide different mechanical parameters, and we just have to be aware of what we are comparing. It's like stiffness. Is it structural stiffness, or is it actually the Young's modulus? And people get easily confused. And not to put only the blame on the biologists, the same is the blame on the engineers who test this. They say, oh, look, it varies by order of magnitudes. But okay, one was dry and the other one was fresh. One was from an old insect, the other one's from. So what did you measure?
Yes.
I fully support that there's a huge many orders of magnitude. I can't say whether it's 5, 10, or 8 orders of magnitude, but the Julian's paper is obviously right, and the values reported, assuming they are true, and I do, it shows the huge variability of the options, and we just have to make sure what are we actually talking about. Not the cuticles probably would be a better way of not talking about cuticle, but cuticles.
Absolutely. Thank you for the answer. Again, I'm asking all the questions and we don't have much time. We can take one or two questions if there is any.
I presumably talk way too fast for several people in the audience. I apologize for that. But when I get excited and talking about insects excites me, I speed up a lot. So
That's perfect. Thank you. I would like to ask another question here too.
go ahead, Hamed.
Because you mentioned the results about fracture testing or tensile testing of the wings, and you have measured the fracture toughness of the wing. If we want to compare the fracture toughness of the wing with any engineering material, what is the closest engineering material that we have to the wing material, wing cuticle? Yeah, sorry.
No. So for the tibia, the fracture test, it is comparable to antler. Well, I have to think which one is the biological composite for that.
No. I mean engineering material. Do we have any?
I have to. I'm not actually sure. I would have to look this up because I'm usually thinking biological materials.
But then my question would be, you know, this sometimes this discussion between colleagues whether cuticle is a real is a brittle material or is a dark hard material. Can we really say something, or that also really depends on
That's again the point being that some parts of the cuticle are certainly very brittle. If you look at the mandibles, they're like one general feature of most of the things that we know of, it has an excellent combination of high. The work of fracture is very high for those of you who know what that is. So it's actually it's very good. It can take cracks to a large extent, but it still has a high stiffness. But of course there are parts of the cuticle, the membranes, which have a very low stiffness, so they are more ductile, and there are other parts, the claws or anything, the mouth parts, which will behave way more brittle. So again, if some engineer asks you whether it's a brittle or ductile material, you say depends on which part you look at. If you want me to show you a ductile material or cuticle, I'm going to show you where to look at the insect. If you want me to show you a brittle one, I'm going to show you another part of the insect. So it's and this is again the awareness, I think, that counts, like not to oversimplify it.
Okay, I see that. And again, I remember that you and David were working on or you had a PhD student working on the any evidence of healing in the wings. Did this led to any specific result? Did you find any evidence for it or no?
Yes, so first of all, this is a project we're currently starting again with a DFG fund. I'm very happy to do this. So healing in cuticle is something we're currently working on. With Owen, the student, and I've been supervising with David together, we did one of the first systematic studies on healing of cuticle. And we found that, first of all, it's able to heal, which to some extent. So usually cuticle can clog itself. So you cut yourself and it just clogs. It closes the incision with some cells and some material so the insect doesn't dry out. That was known. But what we also found, when Owen founded his PhD, is that there can be some targeted deposition of cuticle. So parts which actually, so it's not just like, oh yeah, I'm going to close it, but some parts which are under more stress presumably have a different type of repair response. So this is extremely exciting. What we think, which again shows that it's a different, it's the same concept in biology that has evolved again, like with your bones. Bones and cuticle are like as far away as you could possibly imagine. And here, bones have invented that, or found it, know where I get money. And cuticle basically has, apparently, we're not entirely sure, work in progress, found like a similar solution of targeted deposition where there are certain amounts of stress.
But within the wing, is there anything?
In the wing?
Yeah.
And we only tested in the tibia, so the wing is something.
Okay, I see.
Christoph, and if you ask me again in 3 years' time, probably Christoph and Jonas will tell you more about it. So Christoph is looking at the micro damage, and Jonas will be looking at fatigue. So we will be able to include both of these projects include a significant proportion of healing.
Okay, perfect. Thank you. Again, if there's any question, please ask because we are 10 minutes out of over the time. I don't know whether Shime is still around, but I assume that we should be slowly saying goodbye to all the audience. So again, if there's any question, please quickly ask your question. Otherwise, we should be saying goodbye.
Thank you very much.
Professor Dirks, can I ask a question?
Oh, yeah, please go ahead, please.
Okay. My question is from your topic, and I think my experience here is that when we trying to transfer the functions from insects to the reality. For example, we want to create some like insect inspired robot, and the result is that the robots we have made is not that looks like a feasible or flexible like that real insects. So what is the reasons? What are the reasons? Or do you have some suggestions when we're trying to make the transfer from the insect skill to the engineer skill on robot?
Well, that's something which I've been. I started my career in working at the University of Bielefeld. They're building some exciting insect-inspired robots. At that time, they built the robot also to learn about biology. So to sum it up, not to give a too long answer, the insects, the six-leg walking gait is something obviously that has many biomechanical advantages. So regarding stability, regarding speed, and so on. The problem that has been going on for many years, and I think this is being solved more and more, is the control of these legs. Because if you have 6 legs, they all have to react to the ground. Because you don't want to have a static movement pattern. This has been tested for many years. You just, okay, I can easily use Lego to build a 6-legged robot walking. But as soon as it encounters any problems, then it's basically helpless. So how do insects do that? And they use very limited hardware to solve this problem of stabilization of walking. I think this is the exciting part. Insects can do this without a supercomputer that scans the entire 3D surrounding of the environment and builds the best path. They just keep walking. And I think this is one of the advice which I learned by my professor, Professor Kruse, is let the physics do the work. Mm-hmm. So sometimes it's like self-stabilizing systems where you say, I don't need high-end control, real-time computing power to measure the blah, blah, and servos. And if you build a system that's mechanically stable and mechanically able to do things by physics, using gravity, things which can't fail, that can solve many of the engineering problems. And then you only need a little bit of biological tuning here and there. So often you see biological inspired robots which are totally over-engineered, right? They have like supercomputing power and light vision with 3D LiDAR, blah, blah, blah. Insects don't have that, and they still work because they clever use limited hardware. And I think that's the beauty of it. And you can even take off a leg and it still works. Try this with a fancy Boston Dynamics machine. Many of them will just fall over. Not all of them. There are people working on that, but that's the advice or the ideas that I have.
Okay, so it means that we have to consider more about the physical mechanisms when we try
I would say, like the exoskeleton, something the hardware, like the passive hardware. How does the thing look like? Where is its center of mass? How does it is it stable by itself? And not, I need muscles and I need to control real-time measurements. And this is all like energy consuming. This is all prone to failure, feedback mechanisms, and so on. Engineering-wise, certainly feasible. With computer power is cheap. We can do this. You can now build insect-inspired robots which really look like insects and they behave like insects. But this is not biomimetics. It's just copying something that looks like biology, right? And say, it's a nice robot. It's a cool robot. I'm not blaming engineers. It's a fantastic piece of engineering, but it's a cool robot. It's nothing to do with insect. It looks like an insect maybe, but it has nothing to do with an insect anymore. Which can be good or bad. It's a decision of the scientist. But from the biomimetics perspective, they say, yeah, this is a great robot, but it's not biomimetics.
Okay, so it means the mechanism for the insects is much more complex that we could
it's actually easier
to apply to the robot. Yes. Okay, thank you very much.
Today's robots are, I think, way too complex for insects. We think how many neurons does an insect have and how many transistors or wires does a modern robot have? Like robots are far more complicated and complex than most of the insects. Insects are just a very, very clever use of hardware.
Thank you very much.
Thank you very much. Thanks, Professor.
So I assume that we should be slowly saying goodbye. Thank you again, Jan-Henning, for the very beautiful talk.
It's a pleasure.
And for the audience are more than welcome to write you. I assume it's fine.
Yes, go ahead.
If there's any more question. And yeah, I will be talking to you hopefully very soon.
Hopefully very soon. Thank you everyone for the invitation, Hamed. It's great to see you again. Everyone else, have a great evening, night, whatever it's time it is at your place. And I'll looking forward to staying in touch.
Thank you very much, and thank you,
Bye. Thank you.
and others,
you.
for
Thank you.
organizing.
Bye.